Hi, hello, welcome back to Physionic. Today's episode is going to be dissecting two different studies to get to the bottom of one question that you may want an answer to, and then a second question that you didn't even know that you wanted the answer to, but I'm going to answer it anyway. So let me unpack that a little bit as we introduce the topic. So we're going to be discussing how caffeine affects our lifting performance, resistance training performance, any sort of high burst activity performance. And so I'm going to answer that, but I'm also going to answer a particular question that, like I said, you may not even know that you wanted the answer to, in that there are a lot of genetic variations in terms of how we metabolize caffeine. And those genetic anomalies or those variations can lead to differences in the results that we see from caffeine's impact on lifting performance and ultimately the ability for us to build muscle from that improved lifting performance. So I'm going to cover a paper that's going to give us the answer to the first question: does caffeine improve our lifting performance? Which I guess I'll quick jump to what I've got listed here. So does caffeine improve lifting performance? And the second question, do your genes matter when it comes to how caffeine impacts lifting performance? If you're not familiar with who I am, my name is Nicholas Verhoeven. I guess I should quick add, I'm also going to explain the physiological mechanisms of how caffeine employs this effect, and through a number of different mechanisms. Okay, that said. Now, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a doctoral candidate in molecular medicine, and I hold my master's in exercise physiology. And I have a decent amount of nutrition science background as well from undergrad. So with those credentials out of the way, let's go ahead and actually jump into the real science, not just credentials. Okay, so we already covered the question that we're going to be examining. Here are the two papers that we're going to be looking at. So the effects of acute caffeine ingestion on resistance training performance and perceptual responses during repeated sets to failure. So this is going to answer, does caffeine have a benefit for resistance training or lifting weights? And the second paper is a little more complex, but I'm going to be focused specifically on the resistance exercise portion, but it's looking at this gene, which is known as CYP1A2, and I'll explain that once we get to it. But first, I'd like to just break down this first study. And this should be relatively simple. It's not going to be overly involved. So I don't want you to feel like, oh, wow, I don't recognize some of these words. That's okay. It's not going to matter because I will explain them to you. All right. So a real basic setup on the study design for both of these studies. So both of these studies followed a similar design. So it's not like we're going to have to go over multiple study designs. They recruited individuals. I think they were all pretty young, in their 20s, maybe late 20s for one of the studies. So, but anyway, quite young individuals. Then they had them come into the lab where they performed a 1 repetition maximum, which means that the participants had to incrementally increase or jump up in weight until they were doing an exercise and they only did it for 1 repetition. So they could only successfully accomplish 1 repetition with good form. Okay. And that would be a 1 repetition maximum because they can't do any more weight than that. So it is the maximum weight that they can do for 1 repetition, hence the name. Now, one study did it for the bench press and the leg press. So we're going to get lower body and upper body measurements. And the other one just did it for the bench press. Is that probably going to matter? Probably not, but there you go. There's the information. Now, after those participants come in on a separate day, so they're given time to recover, and on a separate day, then they are they're given pills essentially, or sometimes a liquid, and they're offered this liquid or pill that they don't know the contents of, and they consume it like 30 to 60 minutes prior to the actual testing. So this first test is just to determine the weight that that they should be lifting, the weights for the real measurement, if you want to call it that. So caffeine was consumed at, I believe, 3 milligrams per kilogram of body weight, and maybe the other study did 5 milligrams, but definitely 3 milligrams per kilogram of body weight. And then they had an equal amount of just like sugar found in the placebo. Admittedly, to be honest, I suppose they do that because they want to mask the taste, but in all reality, I typically prefer not to have any sort of sugar in the placebo, unless I suppose maybe if it gets combined with caffeine, but then you have to assume that caffeine doesn't have a reaction with the sugar, leading to decreased or increased absorption of sugar. But it's a small point, but I just wanted to throw that out there. So the placebo does not have caffeine, and the caffeine group obviously does have caffeine. But they don't know what they've consumed, or theoretically they don't know what they've consumed. And then 30 to 60 minutes later, they undergo this measurement where they do a bench press at 80 to 85% of the 1 rep max. The reason why there's a range here is because one study did 80%, which is the first study that we're going to be looking at, and then the second study did 85% of 1 rep max. So that's where this came in. So they've got their 100% weight that they can't lift any more than that, and then they just bump that down by 15 to 20% to the 80 to 85% level, and then they just tell them, do as many repetitions as you can possibly do on this exercise at this weight. We've already predetermined the weight based off of your 1 rep maximum. And that's it. That's the study design. I'll touch on a little bit more when we talk about the genes, but for now, let's just leave it there. Okay, so this is the first study, and there are some pretty intriguing results here. Nothing altogether shocking, but definitely intriguing. So let me start off here. If we look at, this is, I should have labeled these, but this is the top 2 here are bench press, and the bottom 2 here are for the leg press. So we've got an upper body exercise and a lower body exercise. And again, they're being either given the placebo with no caffeine, so that's in these white bars, or in the black bars, they've been given caffeine. And then they have these individuals do 3 sets of the exercise. So it's not like they're just doing 1 set and then they're done. They do 1 set, then they get, let's say, 1 minute of rest time, and then they have to do the next set at the same weight, and then they get 1 more minute of rest, and then they do another set, something along those lines. But ultimately they finish with 3 sets of total work. So the total amount of work that they accomplished with caffeine was higher than what they accomplished with the placebo. So this already indicates that there's a benefit of caffeine consumption. But what's intriguing here, the second piece that I think is really intriguing here, is the fact that the effect only persists for the first set. Now, these were done, I'm pretty certain that these were done within the same session. So this indicates that, well, one, it indicates that, of course, they're going to get progressively more tired. So, on the placebo, they accomplish maybe let's say like 13 repetitions, something like that. And with caffeine, they were able to accomplish maybe 14 repetitions. So it adds an extra repetition on your first set. However, when we look at then the second set, we see that effect no longer exists. So that the only benefit that we see is only on that first set. So once being tired kind of weighs in, then you ameliorate the effect of caffeine. Although ameliorate is technically a positive word, but regardless, you know what I mean. So then if we look at the leg press, we see the same effect, which is intriguing because what it also tells us is that if these were in the same lifting session, which I'm pretty sure they were, then that means that you don't have to pick or choose which exercise you want that caffeine benefit from. If you do bench press first, and then you do another exercise that works a different set of muscles, the caffeine is still going to have a positive effect. It's just that it's only going to have a positive effect for that first set. So you're going to feel like you can get, or you will accomplish one more repetition from caffeine if you consume caffeine at the amounts indicated. And of course, then the total volume or the total amount of repetitions accomplished were again increased with the caffeine. So lower body or upper body, at least by these general measures. If you want to get more specific, then you might have a critique against that. But lower body or upper body, you see a benefit of caffeine consumption. Okay, so that's pretty interesting. However, now we can introduce this other factor, the question that you didn't know even existed. So, for a bit of background, caffeine here, the caffeine molecule, let's say you consume caffeine, let's say you have a cup of coffee, and that caffeine then enters your bloodstream. From there, what happens is that to get rid of caffeine, your body uses your liver, so these cells called hepatocytes that are found within your liver, and they can express, they have the genetic code for an enzyme, or a gene, I should say. So here's the genetic code for a particular gene in this genetic code that is called the CYP gene, CYP1A2 gene. And ultimately, this holds the information for an enzyme, which is a protein. So this enzyme binds to caffeine all within the liver, all within the cells, although I don't have the cells displayed, but this is all within the cells, and will then convert caffeine to other metabolites. The primary one, and I'll show you an image of this in a little bit, the primary one being paraxanthine. So it sort of, quote unquote, inactivates caffeine. It turns caffeine from caffeine, the caffeine molecule, to paraxanthine, as well as two other metabolites. But the main one is paraxanthine. The name of the metabolite doesn't matter that much. The point is that you're converting it from an active caffeine to an inactive caffeine. And I put quotes around inactive because really these metabolites can also have other effects but I'm just saying from a caffeine standpoint, it is no longer caffeine, therefore it is inactive as caffeine. Okay, so that's how it normally happens. That's how it happens in almost every single individual. Well, every individual that has this gene has the ability to generate this particular enzyme and inactivate caffeine. Now, of course, if you have faster reading of this gene, then you may have more of this protein, for example, of this enzyme, which is, if you're interested, called cytochrome P450. This enzyme, and if you have more of it, then you will be metabolizing caffeine at a much quicker rate. However, there are mutations, variants, single nucleotide polymorphisms, those are all three the same thing, okay, that can change the effectiveness of this enzyme, okay? So if you have this gene, let's say this is the entire gene for this particular enzyme, this gene is constructed by what are known as nucleotides. You know how proteins are made up of amino acids? Well, this gene is made up of nucleotides, which have a different structure than amino acids, but they are the building blocks of genes, okay? So deoxyribonucleic acid, you may have heard of that, DNA, right? Well, those DNA strands are made up of nucleotides. And so here we've got an image, a really basic image that I created where this blue line is a nucleotide and this red line is a nucleotide, and they kind of bond together in such a way. Now you have 4 different types of nucleotides that are repeated over and over in particular organizations. So you might have an A, then a C, then another C, then another C, then an A, then it just continues down this strand, and the order determines how it's read. and what information it holds. So if it's in a particular order, then it will read this particular gene and therefore the instructions for this particular enzyme. Okay, so fair enough. However, some individuals, as I mentioned, have a variation in that one of these nucleotides is not the quote-unquote normal nucleotide, as in there's a substitution with a different nucleotide. So that substitution, so typically fast metabolizers of caffeine have an AA genotype, meaning AA as the nucleotides make up this protein, this enzyme. However, slower metabolizers have an AC, so there's a difference there in the nucleotides. And then you have another group that is the CC genotype, and those individuals have really slow metabolism of caffeine. Now, if you choose to tune in to my endurance, I'm going to be covering all of this again for endurance specifically. So endurance sports like running, cycling, things like that. There's something very unique about this enzyme in that it truly affects endurance athletes quite a bit. So, and there's specific information related to that on how you should take caffeine as a result. So the question is, do these polymorphisms, do these mutations, do these variations in the gene affect the enzyme to such a degree that the speed of the metabolism of this enzyme, of caffeine, does it affect lifting performance? Okay, I realize that was a long-winded way to get to that question, but truly, I could have just blasted through this and just told you, yeah, there's a gene. But, you know, you need background. You need to understand what this is before we get to the results. Okay, so hopefully that's clear that everybody has this gene, but there are slight variations to that gene. Some people's variations lead them to have a faster metabolism of caffeine, and some people have a variation that is slower in the metabolism of caffeine. Okay, all right, let's move on. So now I made this graph based off of the data that they had in the table that they had. The table is just absolutely massive. It's so many different values, so many different things. Most of them don't change. So I thought to myself, you know what? Let me just create a table. Or let me, no, I'm not trying to create a table. Let me create a graph that's easy to read. Hopefully this is easy to read. Okay, so here we've got our no caffeine condition with the AA genotype, which means the fast metabolizers of caffeine. Here we've got caffeine consumers with the AA genotype. Okay, so here they've consumed caffeine, here they have not consumed caffeine. Here again, no caffeine, and here's with caffeine, but here's the AC. So remember, there's the AA, AC, and CC. And what these researchers have done is group the AC difference or variation with the CC variation into one group. So the slower metabolizers of caffeine versus the fast metabolizers of caffeine. And they want to know if there's still an effect. So let's look at this. What we see here is that, and this is at the 85%, so the last one was 80% of 1 rep max. Here's at 85%, that's why the repetitions are lower. So here, let's say they hit about 7. If I go back here, remember at 80%, they were hitting about like 12 or 13. So 85%, they were hitting 7 repetitions. And then when they consume caffeine, they do see a bump upwards of, let's say, to about 8, something along those lines. So there is, again, this corroborates the previous data, also showing that there's an improvement with caffeine. So now we have 2 studies that show that caffeine improves lifting performance. Now, if we look at the slower variants, the people with the slower caffeine metabolism, we actually see a similar effect. So we see that because we're comparing these two and we're comparing these two. So with no caffeine, we see that they achieve maybe 7.5 repetitions, something like that. Remember, these aren't going to be whole numbers because you're averaging a ton of people into one number. But on the other hand, when they consume caffeine, they were getting around 8.5 repetitions. So they still experienced the benefit from caffeine. So this all implies that there is no effect of how fast you metabolize caffeine. Now, if you are interested, however, you will find that that is not the case when we're talking about endurance athletes. So again, if you're doing any long distance exercise, that is not the case. I will also add, there is a stipulation that I must tack on here, in that the researchers grouped, as I mentioned, the AC individuals with the CC individuals. This could lead to improper results. I mean improper in that, let me go ahead and go full screen real quick. What I mean by improper is that from the other studies that I've looked at, again, those I'll be reporting on for the endurance athletes, they had the separation of CC and AC slow metabolizers, along with the AA fast metabolizers. What they found is that there was a difference between the AC and CC. So if you group them all together, that suddenly dilutes the effect that you would normally have. So the fact that they group them together could mean that you are not seeing an effect between the AA and the fast metabolizers and the slow metabolizers, because potentially the AC group is seeing an effect and the CC group is not seeing an effect. Hopefully I'm making sense here. Let me try to rework that. So it's possible that the AA individuals have the same positive benefit as the AC individuals. So the AC individuals also see a benefit from caffeine, just like AA individuals get a benefit from caffeine. However, it's possible that the CC individuals, which are different from AC, could not see a benefit from caffeine. But we don't pick it up because we're mixing together a group that does see a benefit and one group that doesn't see a benefit. And because the CC individuals are so few and far between that they only make up about 10% of the population, so you need to sample a lot more individuals to actually get enough people into your study, they skewed the sampling, they skewed the group to be more AC individuals. So hopefully that's making sense. So if you have, let's say, 12 AC individuals and only 2 CC individuals, the 2 CC individuals could see no benefit of caffeine, and the 12 AC individuals could see a benefit from caffeine. So what's the overall effect? That there's an improvement in caffeine, because the 12 will drag along, statistically speaking, the 2 individuals that did not see a benefit. So I'm just throwing this out there. I am not saying that that is necessarily the case in this situation, but that was the case for the endurance athletes. So it would have been nice if they had still increased the sampling for this particular study so that they could have separated the AC from the CC individuals. Okay, that was long-winded. I realize that. And maybe you got it immediately, and maybe you still don't get it. I'm sorry. I'm doing my best. But based off the data currently, assuming that the factor that I just mentioned is not a confounding factor, which we absolutely cannot say at this time with this study, what this implies is that genotype, so if you're a fast or slow metabolizer, as I said, there is no difference in the benefit of caffeine. All right. Okay, I'm going to leave it there. Let's go ahead and look at 2 of the mechanisms, potential mechanisms for why caffeine has this positive effect in muscle. And okay, so I realize that this looks absolutely absurd, but don't worry, I'm going to walk you through this. All right, so I mentioned in one of my other videos where I dissected the endurance. So there were added mechanisms for the endurance improvements that are not going to be the case for skeletal muscle in terms of burst exercise or resistance training, the type of high intensity training. So let's start here. Caffeine enters the bloodstream, it's in the bloodstream, and then it can bind to the adrenal glands. And then from here, the adrenal glands will secrete a hormone known as epinephrine. So this epinephrine can then affect all kinds of different systems in the body, but if we just limit it to skeletal muscle here, epinephrine can bind to skeletal muscle, and this is what we're zooming into. So this is all in skeletal muscle right here. So adrenaline, same thing as epinephrine, just a different name, can bind to these beta-2 adrenergic receptors or adrenoreceptors. The same thing. People use all kinds of different names for them. And if you've taken a muscle physiology course, then you recognize a lot of this stuff already. But assuming that you don't, I'm going to give you a brief rundown. So adrenaline or epinephrine will bind to this receptor on the outside of the muscle cell. So this is the cell membrane right here, so also known as the sarcolemma. So the sarcolemma will go deep into the muscle and then it'll come back up here, and these are called T-tubules. Okay, I'm not going to go into the structure of muscle. It's not that important. The point is that once epinephrine binds, that there are a bunch of changes that occur within the cells. So you have this organelle, this factory, this section, this storage house within your muscle cells. So underneath the membrane of the muscle cells, you have this storage site that stores calcium. And the reason why that's important is because calcium, when the muscle cell is activated, it will release a ton of calcium into the cytoplasm or cytosol of the muscle cell, which is kind of the open area of the muscle cell. Now, in this open area, that's where you have the contractile units, or the different proteins that have to integrate with one another to actually lead to the shortening or the actual contraction of the flexing of your muscles. So calcium has to bind these structures to activate them. This is a really simplistic explanation, but if you have calcium release, then you have muscle contraction. Okay, great. Where epinephrine comes in is that it will activate a protein known as cyclic AMP. And there's a number of other proteins upstream of this, but ultimately it leads to the activation of a protein known as cyclic AMP, which then will activate protein kinase A, which is another protein. Again, you don't need to know exactly this, but I'm just pointing out the names for these, because eventually protein kinase A will then activate these receptors known as dihydropyridine receptors, and also just calcium channels as a whole. So if it's the calcium channel here, so known as the ryanodine receptor, or this one right here, which goes from the outside of the muscle cell into the muscle cell. This one is already in the muscle cell. It's just compartmentalized in this particular storage site called the sarcoplasmic reticulum. So either way, pKa will modulate or modify these receptors by adding what's known as a phosphorylation tag. So once this tag is here, it becomes more active. So more calcium is released from the sarcoplasmic reticulum as well as from outside of the muscle cell to allow more calcium to flood in here and then to allow more contraction. Now think about that. If you have more contraction, what does that mean? That translates to generally more force output, and more force output is more ability to move weight, so move resistance. And that's where we get the idea of resistance training. On top of that, another thing that happens is that pKa can also activate the ability for the cells through ERK, which is another protein, or through pKa to allow greater efflux and influx, or the exchange of different ions like sodium, chloride, and potassium. Why this is important is because the electrical activity of the muscle cells is super important in that it allows all of this to happen. So the more of this exchange you have, the faster the exchange, the more often you can contract that muscle cell, the more often you can activate that muscle cell. And this also extends to this protein here called SERCA, which actually reabsorbs the calcium. So the calcium is free floating, it's been released. You have the activity of increased contraction, but for the muscle cell to be reactivated, for it to contract again and again and again and again, like a twitch, the only way that that's possible is if this SERCA repumps calcium from inside the muscle cell back into this storage site. So it needs to be resequestered into this storage site. So with pKa, the phosphorylation of SERCA, and through other mechanisms as well that aren't displayed here, you see this increase in the activity of SERCA. So ultimately, what that means is that not only are you promoting the release of calcium into the muscle cell to increase contraction, but to relax the contraction, because when you flex, right? When you flex, okay, you flexed, now you're stuck. You got to find a way to be able to relax again and then flex again for the next repetition. And really on a microscopic scale, it goes way beyond that. The muscle cells are constantly twitching. I mean, way more than what I can display here. It's just thousands of times they're twitching. And the only way that they're like ratcheting your arm towards the point that you want it to be at. So for that to happen, you have to have calcium that's also released. So that's one mechanism. And the second mechanism is once that calcium is released, you got to pick all that stuff back up and put it right back to where it belongs. And then for it to then be released again. You can't just have it sit out there, because that does not, you need to pull the calcium off to have the muscle cell be able to relax. And then for the calcium to be re-released, for it to bind again, and for you to have that contraction again. And this has to happen thousands and thousands of times. So that goes back to epinephrine binding this receptor and having all these intracellular changes promoting this exchange of calcium and the exchange of ions to allow for greater excitability of the muscle cell. Hopefully that made some sense. If it didn't, listen to my muscle physiology lecture that I did for undergrads. I go into a little bit more, or significantly more detail in that, but I could go in even more detail than what I just went into. Okay, all right. So this whole system is possible. There's also some evidence that caffeine can directly have this effect on increased calcium release, but the evidence is that you would have to take like toxic levels of caffeine for that to happen. So it seems unlikely that it's going to be a major contributor, but this might be a greater contributor. And this also applies to the heart and smooth muscles as well, but in different ways. Okay, the final mechanism that I wanted to briefly discuss as well is to talk to you about, okay, so here we've got our caffeine molecule. Here's the structure of caffeine. And as I mentioned, when it's metabolized, it gets turned into 3 different metabolites. So one of them is paraxanthine, which is 84% of the metabolite production. And initially what we thought is that paraxanthine is this inert molecule, that you produce paraxanthine and then your body just gets rid of it. You just excrete it. However, that is not the case, because paraxanthine, once it's released into the bloodstream, can actually lead to vasodilation, meaning that you have increasing opening of the vessels, of the blood vessels, and this allows more blood flow through that area. Now, if this could have an effect, to be completely honest with you, I think that it's probably unlikely, because the results would indicate otherwise. Let me walk you through this. So when you have fast metabolism of caffeine, then you're producing a lot of paraxanthine, right? Because you're metabolizing caffeine very quickly. However, if you have slow metabolism, you'd be producing a lot less of this paraxanthine. I mean, in the end, you produce the same amount, but the speed at which you're producing paraxanthine is going to be quite a bit slower in the slow metabolism group, of course. So is this likely a factor when we're talking if there's no difference between the two? Probably not. So this is probably more of an endurance athlete effect than it is for weightlifting or resistance training of any sort. But still, maybe it could have an effect on recovery or something along those lines. So I just wanted to throw that in there because I thought it was interesting. All right, finally, here we are. So in conclusion, caffeine improves lifting performance, thereby increasing the potential for muscle growth. Let me jump to this real quick. So why do I say that? Lifting performance, sure, you can lift a weight one more repetition or two more repetitions, something along those lines for your first set, as we discussed. That's fine. But how does that actually translate to more muscle growth? Well, it translates to more muscle growth because all these studies were done to failure. So we're talking about really like high intense lifting sessions. We're talking about you're not just lifting and then just like you could do like 10 more repetitions, but you decide, I don't feel like it, I'm just going to rack the weight. No, these people are lifting to the point where they can no longer lift. So the intensity is way up. Great. That's what you need for muscle growth. But the second thing you need is volume. So you need the total amount of work done, the total amount of weight that you've lifted. So let's say you're doing 3 sets of 10 repetitions. That's 30 repetitions at a particular weight. Well, if you can do an extra repetition or two with every single exercise that you do for the first set, and you do this week in and week out, that means that the total amount of volume that you do is going to be overall starting to pull away from when you don't consume caffeine. So this is the way that you can increase the potential for muscle growth. Now, the reason why I say potential is because your nutrition becomes a major factor in the actual maximizing the stimulation that you added for muscle growth. However, genetic background of the CYP gene may not matter, slow or fast metabolizers, which is determined by the CYP gene. Now, remember, there's that big caveat that I added when we discussed, when I went on this whole rant discussing one of the potential confounding variables here. So that's something that we'll have to look into in more detail in future studies. Okay, hopefully you learned something, folks. If you got to this point, congratulations. I didn't think it was going to last 40 minutes long, but it did because there's a lot to explain. But even on something as simple as caffeine. But hopefully you learned something, and with that, I'll hope to speak with you in the near future. Have a good one, guys. Bye.